Combination strategy for bioremediation. Bioremediation is the process of removal of environmental pollutants using biological systems including plants or microorganisms. If we are using plants, it is called as phytoremediation, and if we are using organisms, microorganisms, it is termed as microbial bioremediation. There are various processes which are involved in these remediation techniques. So here we are focusing on two main processes, that is bioaugmentation and biostimulation in bioremediation. A combination strategy for bioremediation involves use of multiple complementary approaches for the cleanup of contaminated environments more effectively than a single method alone. This is often necessary because different pollutants, environments, and conditions require tailored techniques to maximize degradation or detoxification. So, if we combine more than one processes together, we might get a synergistic effect if we study the processes properly. And then the synergistic effect can remove the pollutants more effectively from the environment in less time. Bioremediation uses a metabolic capability of microorganisms to degrade or transform environmental pollutants into less harmful substances. Among the various strategies employed, the synergistic use of bioaugmentation and biostimulation has emerged as a potent approach to enhance the efficiency and rate of contaminant degradation, especially for complex and recalcitrant pollution scenarios. These methods can be used for removal of heavy metals, dyes including azo dyes and other synthetic dyes, and hydrocarbons. Bioaugmentation. Bioaugmentation is the process that involves the deliberate introduction of exogenous pollutant degrading microbial strains or consortia, that means a mixture of different organisms, into the contaminated environment to accelerate the biotransformation of specific contaminants. So, even if pollutant degrading organisms are not present at the site of pollution, we can add some organisms which have shown the capability of degrading such pollutants. We can add such organisms at the site of pollution to degrade the pollutants. This technique is particularly beneficial when the indigenous microbial community, that means the organisms which are present already at the site of pollutant, they lack the necessary catabolic pathways, that means the pathways which are required for the breakdown of pollutant, or sufficient biomass to effectively degrade the pollutant load is not available. The introduction of microbial strains are typically isolated. These strains are typically isolated. They are characterized for their metabolic capabilities to utilize the target pollutant as a carbon or energy source. We can also add genetically modified organisms to such sites. The microorganisms which are used for bioaugmentation are oftenly selected for their genetic and enzymatic repertoire that enables the degradation of xenobiotic compounds such as polycyclic aromatic hydrocarbons, chlorinated solvents, or heavy metals through the biotransformation mechanisms, so that these organisms will take up the pollutants from the environment where they are acting as a pollutant and they are showing some toxic effects, and then they might convert these pollutants into less toxic forms. Bioaugmentation can significantly reduce remediation time frames by enhancing the catabolic diversity and density of the microbial community, thereby overcoming limitations related to indigenous microbial insufficiency or metabolic inactivity. The organisms which we can add at such site may show different catabolic diversities, as we can add a single strain or we can add a mixture of different organisms. The mixture can be of different bacteria or bacteria and fungi together. So all these organisms show the different catabolic diversities and also the density of microbial community increases, which will ultimately result in increased bioremediation of the pollutants. Biostimulation is the process where we optimize the environmental conditions to stimulate the growth and metabolic activity of the native or indigenous organisms which are present at the site of pollution. So biostimulation differs from bioaugmentation in a way that during bioaugmentation process we add the organisms, whereas in biostimulation we increase or we enhance, we support the growth of organisms which are already present at the site of pollution. This is typically achieved by the amendment of limiting nutrients such as nitrogen, phosphorus and trace elements which increase the growth rates of the indigenous organisms, or we can add certain electron acceptors, for example oxygen in case of aerobic processes, and if the conditions are anaerobic or the organisms which are present are anaerobic organisms, we can add nitrate or sulfate, or organic substrates can also be added as they serve as co-metabolites or energy sources. Since many contaminated environments suffer from nutrient depletion due to pollutant toxicity or natural scarcity, biostimulation serves to alleviate these limitations, thus enhancing microbial proliferation and enzymatic activity. That means many of the contaminated sites are having pollutants in high concentration. So sometimes organisms which are present in the environment may not be able to multiply or survive in the required rate. So if we are applying processes like biostimulation where we are monitoring the required conditions for microbial growth and metabolism, then such limitations can be overcome and ultimately the microbial proliferation, the rate of multiplication of these microorganisms and their enzymatic activity is enhanced. For instance, the addition of nitrogen and phosphorus can promote biomass growth, while oxygen injection can stimulate aerobic degradation pathways. Careful calibration of nutrient ratios and concentration is critical to avoid inhibitory effects. Also, if we add all these nutrients in excess amount, it may lead to eutrophication. If we are adding such organisms in the aquatic environments, or if we are adding the nutrients in aquatic environments, and if the nutrients are in excess, then it might stimulate the growth of algae which can ultimately hamper the aquatic ecosystem. Synergistic effects of bioaugmentation and biostimulation. Synergistic effect is the combined effect of these two mechanisms. When combined, bioaugmentation and biostimulation provide a complementary approach that addresses both the biological and environmental constraints limiting biodegradation. While bioaugmentation supplies specialized microbial population with enhanced catabolic potential, biostimulation creates conductive physicochemical conditions that sustain microbial viability and activity. So both these processes work hand in hand, ultimately showing increased rate of bioremediation. This synergy is particularly effective in heterogeneous or heavily contaminated sites where indigenous microbial communities are either insufficient or inactive due to toxic effects or nutrient limitations. Sometimes it may happen that the polluted site will have different pollutants together. So indigenous population may have some strategies to degrade some of the pollutants, but other pollutants might act as the toxic factors. So if we add some organisms exogenously, then those organisms can sequester the pollutants which are not allowing indigenous organisms to grow. The co-application can result in accelerated contaminant removal rates, expanded substrate range, and enhanced stability of microbial consortia. Scientific evidence and applications. Numerous laboratory and field studies have demonstrated the effectiveness of combined bioaugmentation and biostimulation strategies. For example, In hydrocarbon contaminated soils, inoculation with alkane degrading bacteria coupled with nutrient amendments significantly increased the rate of petroleum hydrocarbon degradation compared to either approach alone. Similarly, in chlorinated solvent bioremediation, bioaugmentation with dechlorinating bacteria alongside electron donor amendments. So we are adding the organism which can dechlorinate the environment, and also we are adding electron amendments. Addition of dechlorinating bacteria is a part of bioaugmentation, whereas adding electron donor amendments like lactate or vegetable oils acts as the part of biostimulation. So both these strategies combined have successfully enhanced reductive dechlorination processes under anaerobic conditions. The integration of bioaugmentation and biostimulation represents a robust bioremediation strategy that maximizes microbial degradation potential by both supplying specialized degraders and optimizing environmental factors that might limit the process of bioremediation. This dual approach offers a flexible yet effective framework for remediating a wide range of contaminants. With ongoing research focused on optimizing microbial consortia, nutrient formulations, and delivery methods to further enhance bioremediation outcomes. So, all these methods together can increase the efficiency of bioremediation, and we can clean the environment without production of any harmful molecules.